thank you for introducing. And first of all, I'd like to thank organizing committee to give me this valuable um, chance to talk about my research work. Today, I want to talk about abscisic acid and the mitogen-activated protein kinase. Okay, this is my profile, but I will skip them. Oh, firstly, I want to introduce my collaborator. This is Professor Namori, and this is Takuto Yasufuku. Today's topic is working with him, so I introduce him. Okay, this is outline of today's talk. Like title, my I have two keywords. The one is map kinase cascade, and the other is ABS signaling. So I want to introduce two keywords at first, and then I want to talk about my research. Okay. Oh, this is overview of map casing and pathway. Mitogen-activated protein kinase is minimally composed three kinds of kinase named MAPK and MAPK kinase, MAPKK, and MAPKK kinase, MAPKKK. And too many K. I'm sorry. This cascade is composed, conserved in all eukaryotic cells and have an important role in signal transduction. And these stress or growth signals activate the upstream MAPKK, and the activated MAPKK phosphorylate MAPKK, and then activated MAPKK phosphorylates its downstream MAPK, and activated MAPK phosphorylate some target gene like transcription factor and regulate gene expression and enzyme activity. And these signals connect to the cellular response. So in Arabidopsis genome, 18 MAPKK and 10 MAPKK and 20 MAPK genes are in the genome. And it also knows the total protein kinase gene is 1,000 and about 300 gene exist in the Arabidopsis genome. About 10% of the gene is MAP kinase cascade component genes. I don't know, it's the half of about 600 gene is receptor-like kinase, and 10% is MAP kinase cascade. So I think they have a important role in signal transduction. And each stress choose specific MAPKK or MAPKK and MAPK composed specific kinase cascade for each stress. For example, I will show you that representative signaling. We published this kinase cascade wounding signal activate MK1 and MKK1 MPK4. And other group also shows bacterial pathogen signaling also activate MK1 and MKK1, and also MKK4 and 5 and the downstream kinase. And also other group shows cold and salt stress activate these components, MK1 and MKK2 and MPK4 and 6. So many signals concentrate to MK1 and specific MAPKK is activated and the signal is transmitted. And also we recently, dot signal also activate MPK4, and recently we have published these components upstream of cold signaling. Upstream of McKeven signal in cold signal. So I will show next ABA. Oh, sorry. Oh, the other keyword ABA. ABAs also have a lot of function to regulate the developmental stage or stress signaling. These biotic stress like drought, assault, or developmental stage, seed maturation, or dormancy, is induced ABA biosynthesis. The ABA signal is transmitted to these ABA receptor and PP2C phosphatase and SNRK protein kinase. This is called ABA core signal. And then SNRK phosphorylate some target gene to regulate enzyme activity and or transcription factors. So ABA signal is, for example, rapid response is stomata closure is a typical rapid response. And delayed response is okay. So, and also these signal components is involved in ABA signaling, but there is a lot of things to be analyzed. So, next I will talk MAPK signaling. Okay. Involvement of MAP kinase cascade in ABA signaling. These MAP kinase and this MAPKK is reported to involve in the ABA signaling. But now upstream MAPKK is not identified. Recently, we and the other group published the MAPKK 18 and 70 is working in MAPKK in ABA signaling. So to identify the MAPKK in ABA signaling, we analyzed the Arabidopsis database expressed, for example, expressed in ABA signaling, and I found MAPKK 18 is expressed by the ABA signaling. So first work is to analyze the MAPKK 18 in ABA signaling. So I'd like to talk about research one. And we produced overexpression lines of MAPKK 18, and for analyzing ABA signaling, the role in ABA signaling and the physiological role. Today's talk is about this paper. And okay, to confirm the ABA induction of this gene, real-time RT-PCR analyzed. So MAPKK80 we used a 2-week-old Arabidopsis were treated with ABA, and the expression were analyzed for each time. And you can see after 30 minutes treatment, clear induction were observed. And the ABA1, ABAI1 is protein, ABA inducible protein phosphatase, and works in ABA core signaling. And it is also known ABA responsible gene. So to confirm the ABA treatment, I use this gene. And to analyze the protein kinase activity, this we expressed in the E. coli, and without protein were used for protein kinase activity. And we use the MVP for substrate of this kinase. And you can see the clear band of MVP, and also this band is from autophosphorylation of MAPKK18. And we also make a kinase negative mutant, like this lysine residue to substitute to arginine residue. This lysine residue is conserved for the ATP binding, so this mutant cannot bind ATP. So this mutant cannot phosphorylate. So there's no activity here. To identify the downstream pathway of this KKK, we use the yeast-2 hybrid system. And as I told, in Arabidopsis genome, 10 MAPKK gene exists. There is 10 MAPKK genes. So I try to analyze these binding. So you can see the clear blue colony from the binding. So MKK18 can bind MKK3. So the same way, the MKK3 bind to MPK1 to 7C group MPK1. So to summarize these results, MAPKK80 induced by ABA signal and conform these cascade. So next I want to the role of this cascade. We make an overexpression line of this. And this is the vector for overexpression. And to analyze the kinase activity, kinase negative mutant were also overexpressed. And to detect the overexpressed protein easily, fractal is fused to N terminus. So, and overexpressed protein is analyzed IP kinase assay, and we use a 2-week-old Arabidopsis plant and extracted total protein, and this is a fractal western blotting band. In the ABA treatment, the level of band is not changing, but kinase activity were used to was analyzed by use MVP. So clear band was detected in the 30 minutes in ABA treatment. So ABA also regulate this kinase activity. So I will show the phenotype of MAPKK 18 overexpressed. To compare the growth, we choose two lines, each two lines of overexpressed MAPKK 18 and MAPKK 18 kinase negative. So you can see the clearly the growth is clearly inhibited in this overexpressed line, and the kinase negative line is increased growth. So to compare this growth difference in detail, we measure the fresh rate, and also the green color is monitored by chlorophyll content. So after 40 days, this difference is clear. For example, chlorophyll content is lower in the overexpressor, and kinase negative mutant is still green. So, and the seed productivity is monitored, and the seed number is twice in the kinase negative mutant. So, and transgenic plant overexpressing MAPKK18 shows early senescence phenotype, and the growth was inhibited. And the transgenic plants overexpressing the kinase negative mutant is delayed senescence, and then increased biomass and seed productivity. So MAPKK 18 is very useful to change the growth timing and productivity. So this is ABA response of the transgenic plant. We tried two kind of experiment. The one is leaf senescence assay, and the other is water loss rate of detached plant. So in the presence of ABA, the leaf color changes to yellow, and in overexpressed line, this color is severe yellow, and on the other hand, kinase negative is still green. So ABA signal is this phenotype. This line is very sensitive to ABA, and this line is insensitive to ABA. And also, water loss is still remain water this full length, and kinase negative mutant is more water loss. So ABA, in the case of ABA signal, the ABA work to tolerant to drought. So okay, sorry. Overexpression of MAPKK80 enhances the sensitivity to ABA, and on the other hand, overexpression of MAPKK18 can inhibit the ABA response. So, to summarize, MAPKK18 is a positive regulator of ABA signal. So, okay, this is summary of research one. I already talked about this. And the overexpression of MAPKK18 gene affect the ABA signaling and elevated sensitivity to ABA. They show early senescence and drought tolerance. On the other hand, MAPKK18KN inhibit the ABA signal, delayed senescence, and increase biomass and seed productivity. So next, this is only specific to the MAPKK18, or other MAPKK is also same phenotype is important. So we make overexpression plant to other ABA induced MAPKK. So this is the table. Not clear, but these genes are regulated by ABA, and we choose from these two genes are chosen, and one is MAPKK17. The expression pattern is almost the same with MAPKK18, and also bind to MKK3. So MAPKK70 is redundant function with MAPKK18. So okay, I'm sorry. And also we make an overexpression line. So this is phenotype of this plant. And this is preliminary result, but the growth is inhibited and in full length. And the kinase negative mutant is, the growth is increased. And the bolting is also earlier and delayed bolting. So, and also we have published other ABA responsive MAP3K, and also growth induction, and interestingly high salt tolerance. And this is conclusion. The biomass and seed production can be controlled by overexpressing ABA inducible MAPKK. So, but this is in only Arabidopsis case. So, and I don't know why this phenotype is produced, but this, next project is to analyze these mechanisms. But the other plant is more important to agriculture. So that's all. Thank you for your attention.